BY4 mock, it's the June 2015 paper, question 4. So here's a practical that most of you will have actually done, separating the pigments from photosynthesis in a plant. The blue-green pigment is chlorophyll A. State precisely where chlorophyll A is found in a photosynthetic cell. So hopefully you've got your photosynthesis notes and you're going, where do I actually find this stuff? So it's all to do with the light dependent reaction, and then can you find on these diagrams somewhere where chlorophyll A is going to be. So that's just in your notes. Here's a calculation, and it actually gives you the equation there. So calculate RF value of the yellow band and use the table to identify it. So what you need to do is look at these. Where's the yellow band there, and where's the solvent front there? And so with your ruler, measure that, measure the solvent front, put those into here, and how many decimal places should I use? Well, how many decimal places am I given there? And I'm usually going to follow the same style as I've been given. So I'm going to use 2 decimal places there. And that number that you come up with should be extremely similar to one of these in the table. You should be able to identify it. Part C. Some of the pigments were extracted and they've done this chart with them. So they're showing them different colours of light, which equals the wavelength, and they're measuring the absorbance. Unfortunately, the examiner is selling you a little bit of a dummy here with the name of this chart. You should know the name of the chart. You should have your notes there and go, yeah, I've got the charts, I recognise this, and I've got the names there. I'm not just going to copy this axis title there. I'm going to remember the proper name of the chart. Explain the results in the 500 to 600 range, so from there to there. And so we have got very low absorbance of the light. So the light is not being absorbed by these pigments. So where is the light going if it's not absorbed? And hopefully you realise why in this kind of green area. That's why plants, leaves look green, because reflection of the light. Explain the advantage to plants of having more than one pigment. Hopefully you notice from here that these are various pigments. It doesn't tell you which pigments are which. I'm just going to look at this wavelength there. Even though this pigment isn't absorbing much light, these other 2 pigments are. Here, this pigment is still absorbing light, and so on. And so we want to know the advantage to plants. So the first point might be describing the absorbance, the advantage at these different wavelengths of having 3 of these lines rather than 1. And then answer the question, the advantage to plants. What are plants going to do with this light that they've absorbed? So what are they going to do with it? A little bit of theory there. Which process do you know about that's going on that involves this light? So which process is going on that involves that light? What are plants going to do with it? Part D. Use your knowledge of photosynthesis. to explain the role of these pigments. So it's basically saying, okay, these pigments and photosynthesis, what's going on? Well, that is a rephrasing of the question, tell us about the light-dependent reaction. So tell us about the light-dependent reaction. And so all this stuff about light harvesting and light-dependent really is producing ATP. So it's all this kind of stuff, light-dependent reaction, and focus on the question, how do they actually produce ATP out of it? And it's a lot of space actually for 3 marks, so you should get those. BY4 mock paper, which is June 2015. Question 5. Diagram shows a batch fermenter. It's not a continuous fermenter. A batch fermenter used to produce penicillin. So lots of familiar things on that fermenter there. First question. Apart from helping with the mixing, explain why sterile air is pumped into the fermenter. So there's 2 marks here. So I would suggest sterile, explain why it's sterile, and 2, explain why air is actually pumped. Okay, so the air contains something that's needed for a certain process, and it needs to be sterile so that something else doesn't happen. So the examiners reported that it was quite rare for people to get both of those marks. The fungus that produces penicillin requires a supply of carbon and nitrogen. In what form might these elements be supplied to the fungus? So you need to remember a fungus, cell wall, cytoplasm with various things inside, mitochondria and so on. What form of carbon is a living cell, a heterotroph like a fungus, going to need? Is it going to be happy with carbon dioxide or lumps of coal being added? No, it's not. It's going to need something a bit more useful than that. And again, with nitrogen, can you use nitrogen gas out of the air? No, well neither can this fungus. So we need what form of nitrogen is going to actually be used by the fungus. How would the pH be controlled? Okay, so I'm not just going to say the first thing that leaps into my head. I'm going to go back to the diagram and have a look for any clues here. Right, pH control reservoir. Okay, so I'm going to suggest the pH might be monitored somehow, but how is it actually controlled? Well, what am I going to add and when? So I might add some things or some other things depending on what the pH is in here. So suggest what they might add in what conditions. What is the purpose of the temperature control system? 2 marks. Again, go to the diagram. They're trying to give you some help here. So the temperature control system, there it is. So it's monitoring the temperature and then it's doing something. So what you need to do is go, right, this is how it can change the temperature by turning this and putting more or less of this cold water into this jacket. So I would suggest you suggest reasons why this might want to be added and relate any biological processes there. And then if this turns and more or less of this cold water comes in, what effect that's going to have. So use the diagram as well as your biological knowledge there. Here is a graph showing the production of penicillin and the growth of the fungus. So it's important here to realise that the fungus is penicillium and the substance produced is penicillin. Again, some problems with reading the graph, particularly with the scale. How many blocks for how many units of time is a little bit of a mystery. And so here 40, 50, 60, how much is each block worth? Well, there's 5 blocks in 10 hours, so each block is 2 hours. So it's the first problem, reading the scale, and then the second possible one might be, well, which bit of the scale am I actually going to read? Enters the stationary phase. So stationary is not moving. So when is this line not moving any higher? And so you are reading the scale. There is a range in the mark scheme, but it's not there. Okay, so you need to be a bit before that. Why is there a high level of protein synthesis in the first 10 hours of the penicillium culture? So we're talking there. So protein synthesis. You need to think of things that are made out of proteins. What would be a useful thing made out of protein to produce at the start? Explain why no penicillin is produced during time X, time period X. Okay, right, time X, here it is. So what we've got, a bit of a lag and the early log or exponential phase. Those are the stages that we're talking about. And can we answer the question? Explain why no penicillin is produced. So I'm not going to explain why loads is produced over here. I'm explaining why none is produced over here. And so you need to explain why the conditions of growth of this fungus is the fungus needing to produce penicillin, and why is it not needing to produce penicillin. So use a bit of your theory there, and that's 3 marks.